Okay, here we are. So we just finished talking about aerobic respiration, and aerobic respiration, which is what we do, humans, it's what we do and what a lot of bacteria do, is a great way to make energy. It's a very efficient way to extract energy out of things like sugars and lipids. It's the best way, the most efficient. But there are a lot of environments where there's no oxygen. And even though there's no oxygen, there's plenty of carbon and there's plenty of phosphorus and nitrogen, and there's a good temperature and good pH and just the right environment for a happy little cell if it has a way of making energy without oxygen. And we will talk about fermentation at some point, and that is clearly one way that cells can make energy without oxygen. But fermentation is not efficient at all. Fermentation is a very weak way to make energy. And the only time that's going to happen in nature is when there's no better option. So let's get into the better option. So the better option is anaerobic respiration. That's respiration that takes place without oxygen. So remember, we're not talking about breathing. We're talking about putting electrons onto an electron acceptor and using an electron transport chain. That's respiration in the physiological sense. Forget human physiology. This is bacteria, this is archaea and fungi and stuff like that. So anaerobic respiration, let's think about what that would require. Well, it requires an electron donor, an electron transport chain, and then electron acceptor. And so what we're seeing here is different types of, chemolithotrophy. Basically these are inorganic electron donors. So they're not using sugar, they're not using lipids, they're not using amino acids, they're using hydrogen or hydrogen sulfide or iron. This is ferrous iron or ammonia. All of these, I've looked at this stuff for many years, and when I look at these, I think these are reduced. They have electrons that they can give up. So these are electron donors, which means they can send electrons through an electron transport chain if there's an acceptor. Well, if there's oxygen, that's rad, and the ones that can use oxygen are going to win. But if there's no oxygen, they do anaerobic respiration. And anyone who can use nitrate, we're going to see later, this is the second best electron acceptor. Anything that can use nitrate is going to win. And then what we will see is a hierarchy of electron acceptors in nature, and I'll explain what that's about. So again, if we were looking at chemolithotrophy, we might see something like hydrogen gas be the electron donor, and something like sulfate be the electron acceptor. This is a common electron acceptor. The waste product of this is hydrogen sulfide gas, which smells like rotting eggs. And guess where rotting eggs get their smell? They get their smell from bacterial sulfate reduction, from exactly this. Although instead of hydrogen, those would be using sugars or proteins in the egg white. But what they're using sulfate as their electron acceptor, since there's no oxygen inside an egg. The same thing happens in the ocean. There's a lot of sulfate in seawater. And so if an animal, like a fish, dies at the beach, it's got sulfate in it. And the anaerobic stuff, like inside its muscles, as bacteria are breaking it down, they will find sulfate, but they won't find oxygen. So they'll use the sulfate as the electron acceptor, and the waste product will be hydrogen sulfide. So you smell that nasty rotting egg smell at the beach. That's because of sulfate reduction. This is an important process in nature. So this is one example I do want you to know. I want you to know sulfate and sulfate reduction and what that means. I don't care as much about the electron donor. I really don't. Electron acceptors really drive a lot of this stuff in nature. We also have chemoorganotrophy. This is what you might be more familiar with, where there are organic electron donors, like what we use, sugars, lipids, Bacteria can use amino acids, lots of other things. And the same options are available for the most part. Aerobic respiration is great. They're going to win. Nitrate is the second best. If they can use that, they'll use it as soon as the oxygen is gone. But remember, microorganisms are highly specialized. So there are not many that could use oxygen and then switch to nitrate and then switch to sulfate. They specialize in using one or the other for the most part. Cool. So nitrate is the other example I want you to know. So the 3 electron acceptors I want you to know are oxygen, sulfate, and nitrate. Those are the big important ones. So this would be very common in nature. This is called denitrification or nitrate reduction. This is nitrate. And the waste product of this could be, I think you'll see it in a bit, nitrite, which is toxic, or nitrogen gas. It just depends on what the cell wants to do. In this case, so this is the second best electron acceptor. This is nowhere near as good as oxygen. This is not as good as oxygen. So if, like this electron transport chain does not pump as many protons across the membrane as an aerobic respiration electron transport chain. I think there would be 1 enzymatic complex that could pump protons instead of 3. So if you go back and look at our electron transport chain, there are 3 different protein complexes that pump protons as the electrons flow through them. For something like nitrate, there would only be 1. For something like sulfate, it's even worse than that. So they get a lot less ATP per molecule of glucose than an aerobic respiration gives us, or gives our mitochondria. Still, if this is available as an electron acceptor, this is the second best option, and bacteria that can use it are going to win. The other option they have, and lots of bacteria do this is very common, is fermentation. It's growth without external electron acceptors. So there's no oxygen, no nitrate, no sulfate. And they have to make their ATP through substrate level phosphorylation. So if we go back to our central energy metabolism, like what we see in aerobic respiration, like our own mitochondria or E. coli growing using oxygen, or lots of other bacteria using oxygen, this is what they have. They would start with something like glucose, and let's just assume they're using glucose because that's available in nature quite often. They oxidize it and make pyruvate. That pyruvate is then further oxidized to carbon dioxide and acetyl-CoA. That acetyl-CoA is oxidized all the way to carbon dioxide in the citric acid cycle. So each of these oxidations that happens releases electrons in the form of these electron carriers like NADH and FADH2. And if we're doing respiration, these go to an electron transport chain, and they dump the electrons in, and that's the start of the electron transport chain. And the amount of energy we get from our electron transport chain depends on the electron acceptor, because that determines how many proton pumping steps we can put in our electron transport chain. Okay, fine. But what happens is if there's no electron acceptor, we run out of the oxidized forms of these electron acceptors. We run out of the forms that are not carrying the electrons, because these reduced forms start to accumulate and they can't cycle back to be these oxidized forms. And that means glycolysis stops because it can't go forward unless it can oxidize this glucose and put the electrons here. Pyruvate oxidation can't happen unless there's NAD+ to take the electrons from pyruvate, and the citric acid cycle can't happen unless there's these electron carriers to take the electrons from acetyl-CoA. So that's why fermentation becomes necessary. If there's no electron acceptor, all of these cycles have to stop. All of these pathways would shut down. And so bacteria and archaea have elegant things they do in the absence of electron acceptors, so that this is not like a terrible thing. This is just a new situation. So these are the types of organisms that would do fermentation. And they're the anaerobes, the ones that can grow without oxygen. Some of these could also do anaerobic respiration. Yeah, some of them can. Pseudomonas aeruginosa, I'll talk about in a bit, ends up being a facultative anaerobe. It grows really well if there's oxygen, like really well. It's famous for it. But it can also use nitrate. It can do nitrate reduction also. But it doesn't do fermentation. So that one's not on this list because it can't do fermentation. So these are the organisms that do fermentation. This is where you would find those organisms, or what groups they'd be in. It's all the anaerobes. Now remember, anaerobes there are also going to be organisms in these groups that do anaerobic respiration using nitrate or sulfate or other electron acceptors. But this is where you find the organisms that do fermentation. So fermentation is how they balance their metabolism when they can't get rid of their electrons. They have nowhere to put those electrons, so they can't use an electron transport chain. They can't do anything really until they have a place to put their electrons. And so what happens is fermentation requires complex organic molecules that have both a part that acts as an electron acceptor and a part that acts as an electron donor. So pyruvate is an example. What I should say is glucose. So in fermentation, what you'll see is that the electron acceptor part takes electrons off, puts them on NAD to make NADH, and then sends them to so what you'll see is that they will use the part of the molecule that could be an electron acceptor, and they'll put the electrons on that to regenerate that NAD+. And the waste products of fermentation, the ones that you're probably familiar with would be like lactic acid or lactate. Or if you've studied this other stuff, ethanol comes from a type of fermentation also. But in nature, more often, fermentation products are stinky, disgusting smells, because this is what happens in corpses. This is what's happening in your distal colon. Fermentation dominates those environments. And so you get just the worst smelling stuff. But I'm getting ahead of myself. So fermentation solves the problem of excess electrons. And again, I'm not asking you to memorize every example. I want you to be able to explain what's on this slide and what's happening and why the cell is doing it. I do want you to be able to tell me in glycolysis what are these two reactants and what are these two products, or these two inputs and these two outputs. You really should know that by now, or if you didn't, learn it. So glycolysis generates pyruvate and two electron carriers. And lactic acid fermentation, like what happens in our own cells, is the NADH donates the electrons to pyruvate, in the process making lactate. And then this NADH can be recycled to NAD+. So in this reaction between these two, this is one of the products and this is the other product. So that's lactic acid fermentation. This is the simplest fermentation. And why do our cells do the simplest fermentation? Well, it is the fastest. It's the fastest way to keep glycolysis going. And every time we send glucose through glycolysis, we make 2 net ATP molecules. And so as long as we can keep this going, we can keep making ATP, but we also accumulate lactic acid, and that is an acid that drives down the pH, as you know. So this is what we would see in yeast making ethanol. It's the same idea. They're going to put electrons onto pyruvate, but instead of making lactic acid, they make ethanol and carbon dioxide. But the same thing happens. The point is to regenerate this NAD+. Okay, so the real specialists in fermentation are bacteria from the genus Clostridium. This should be familiar to you because this genus makes endospores, and many members of this genus are really dangerous. They make toxins that kill us, or they cause gangrene, which kills us very quickly and makes us rot while we're alive. So Clostridium is a fearsome enemy to have. But not every species in the Clostridium is like a human pathogen. But what they all do is they specialize in eating decaying living stuff, like fermenting muscle tissue in a dead person or a dead cow or a dead sheep. And so Clostridium make the world smell very bad. So one thing they do is butyrate fermentation. They make butyric acid and carbon dioxide. And this would be, they'd use both of the pyruvates to do this. And so all the carbon would end up in the butyric acid and the carbon dioxide. I don't know if you're familiar with butyric acid, but if you buy a bottle of butyric acid, it has the word stench in all capitals with 3 exclamation points. That is literally the standard labeling for butyric acid because it smells so bad. If you spill it in a laboratory, you have to evacuate the building. That happened when I was doing my master's. There was a lab on campus where someone broke a bottle of butyric acid, and the smell is so bad no one could work in that entire building, that 6-story building, until they cleaned it up and flushed out the air. So this is something you would smell coming out of a corpse or feces. This would be a big smell in fecal material also. And then another option they have, if the pH gets too low, they don't want to make an acid as their waste product. So they switch to making some kind of mixed fermentation where they're making several different molecules. I should point out, it's not that important for it's not that important a thing for you to learn, but if you count the number of carbon atoms in pyruvate, or in 2 pyruvates, it's 6. It's the same number that we got from the glucose. If you turn them into butyrate and 2 carbon dioxides, it's still 6. Butyrate or butyric acid, same thing, is a 4-carbon carboxylic acid. If you count these, you get twice as many. You get 12 if you count up all of these. And so when Clostridia switch to doing this as a waste product, they actually use twice as much to make these. So in that sort of one reaction they'll use 4 NADHs and 4 pyruvates. So that's why I have ×2 here. That's not critically important for you to understand. It's just I have to make these things correct when I put them in here. So again, I'm not asking you to memorize these. I'm asking you to remember the big points, like why does feces smell bad? It's because of fermentation byproducts like this, like butyric acid, butanol. And the worst smelling things in nature, well, I'm exaggerating, but the worst smelling things coming out of a corpse are from the Clostridia fermenting lipids. And that's a complex process that even the bacterial physiology books I have don't fully explain. But yeah, if they're fermenting amino acids and fats, the products they make, the waste products, just smell vile. And that's why corpses smell so bad. So on that pleasant note, let's stop this video, and when we come back, we'll go right into a slide that kind of looks again at what you should be studying. I think I've already touched on it, but I do have a slide for people who don't watch the videos. So let's look at that in a second. In the next video, I'll see you there.